Hello, this is Iron Nathaniel Plummer.
I'm Khalil Lajide,
This is Nikia Lawrence.
and our fourth group member, Malik Dockery, would not be able to speak today, but
And
we will be talking about cell adhesion. So, first of all, we're going to talk about this cell adhesion and its importance. So, the cell is the building block of life, and such that the similar cells combine to form tissues combine to form organs, a group of organs carry out the same specific that carry out a specific function combine to form an organ system, and ultimately you have your organism formed. Cells attach or adhere to each other to form tissues by means of cell junctions and cell adhesion molecules. So, cell adhesion plays an important role in the formation of an organism. So, what is cell adhesion? Cell adhesion is basically the attachment of cells to one another or the extracellular matrix to for the purpose of communication, signaling, and cellular proliferation and cellular differentiation. Um, so, cell adhesion plays an important role in the formation of an organism, and you could have homophilic or heterophilic interaction or attachment. The attachment between cells or the cell and the extracellular matrix, as the case may be, could be homophilic or it could be heterophilic. So, homophilic attachment or adhesion is basically adhesion between cells of similar receptors. So, homophilic attachments are specific in their attachments because they only attach to molecules of the same receptor. They only attach to cells of the same cell receptor molecules. So, as we will discuss later in greater details, cadherins are homophilic in their interactions between others with other cells. And next, we're going to talk about heterophilic interactions. Heterophilic interactions are the opposite. They are non-specific in their interactions, and they are basically the attachment with of cells or the extracellular matrix with cell receptor molecules that are different from the cells that are being attached. So, cell adhesion, they like I said before, helps in communication differentiation. Differentiation is basically the specialization of cells to form a specific function. And we have two types of cell adhesion. We have the cell to cell adhesion, which is basically adhesion between cells, and we have the cell to matrix adhesion, which is the adhesion between a cell and the extracellular matrix.
cell adhesion molecules. Basically, it has different types of junctions, adhesive junctions, desmosomes, adhesion junctions, tight junctions, gap junctions, and focal adhesions. Um, the space involved between the junctions. Adhesive junctions are cell-to-cell points or places where cells attach to cells function and cell shape. Um, there's also a gap junction and tight junction. Gap junction is intermediate and attached to one another. Tight junctions help connect epithelius and makes it permeable.
So before we move on, basically the we have four different groups of cell adhesion molecules, and we have now I'm just gonna we have the selectins, the integrins, the immunoglobulin superfamily, and we have the cadherins. So we're going to classify them quickly based on their mode of interaction, like we had explained before, whether they're homophilic or whether they're heterophilic. So the immunoglobulin superfamily and cadherins they have homophilic interactions, and while the integrins and the selectins are heterophilic in their mode of interactions.
All right. So for the cell junction, for the figure we explained before, the figure basically explains functions of the junction and how they act. So basically, hemos, hemo, same, hemidesmosomes,
Hemidesmosomes.
hemidesmosomes Basically, hemo means half, and desmosomes that means connected to basal lamen to lower regions and basal lamen region. And then they have the Canton's belt around the apical region, right here, for the adherent junction. Adhesive junctions, as I said, cell-cell adhesive junctions, and desmosomes, and extracellular matrix.
So this figure right here just basically tries to explain a polarized cell. When I say polarized, I mean like they have different domains, and these domains have different functions. So this shows like the apical region, and you have the basolateral region, you have the basement region, you have the different regions that have carry out specific functions when it comes to cell adhesion.
Here we have cadherins. Cadherins is the single pass transmembrane cell adhesion molecule that is calcium dependent. It has a homophilic cell-to-cell interaction through the extracellular domain. There are three types of cadherins, such as E-cadherins, which are found in epithelial cells. We have P-cadherins, which are found in the placenta. And lastly, we have N-cadherins, which are found in nerve systems. So here in the figure, we have basically the effects of cadherins on cell adhesion. Basically, in the study, cultured L cells, which are fibroblast cells, as you can see, they did not, well, not as you can see, but they do not attach to each other. So when E and P cadherins were added to these L cells, and the cells started to produce these different adhesions, P cadherin, which is in the red, and the green is the E cadherins. Basically, these groups of cadherins basically started to attach to one another. As you can see, the green E cadherins attached with the E cadherins, and the P cadherins attached to the P cadherins.
And then here we can see like the homophilic interaction that we spoke about before. We can see that P cadherins only it came together with similar P cadherins to form tissues of to form a specific tissue ultimately form a specific tissue, and the E cadherins would adhere to other cells expressing E cadherins because they're alike, and to form other specific tissues.
So here we have the structure of a cadherin. So you see that the extracellular domain of the cadherin is pretty much similar to a zipper. So it has like a zip-like structure which extends outside of the cell, which then it attaches to the adjacent cell. So in order for this to occur, calcium must bind and stabilize the extracellular domain in order for the zip structure to occur. Then you have the beta-cadherin protein, which binds to the cytolytic tail of the cadherin, while the alpha-cactin binds to both the beta-catenin and the actin skeleton. The beta-catenin is bound to the P120 catenin. It is an adherent junction that binds the tail of the cadherin to the plasma membrane. So the P120 catenin mediates the cadherin stability by making sure that it is secured to the plasma membrane, and it also regulates its Rho activity. So next we have integrins. Integrins are made of two large transmembrane glycoproteins with two subunits, which are alpha and beta, which form a non-covalent heterodimer. Integrins do not directly adhere to the extracellular matrix. The way that they do adhere to the extracellular matrix is by focal adhesions and hemidesmosomes. Focal adhesions are a cluster of integrins that interact with the microfilaments through adductor proteins. Adductor protein, which is one, is taline that binds to the actin binding protein vincillin. and alpha actin in order to directly bind the actin filaments to the cytoplasmic cell of the integrin. So as you see in the figure, the alpha and the beta subunits in the extracellular portion of the integrin interacts by forming a binding site for the fibronectin. Specifically with the alpha subunit. In the cytosolic portion of the alpha subunit, there is a binding site for the adaptive protein taline, which links the cytoskeleton to the extracellular domain.
So now we're going to try to explain how this links to cancer and metastasis. So we understand how cells grow. So cells grow through the process of cell division, whereby the mother cell divides into two daughter cells. So it's a rapid process. It doesn't take time, and it ensures that cells grow when they're needed in the body. So the body works in such a way that as cells divide or grow when they're needed in the body. So if cells are not needed to be replaced by worn out or dead cells, new cells are typically not needed to be made. So I don't expect my body to make cells when I don't need cells to be made in the body. However, when cells grow abnormally, even when they're not needed in the body, we could actually call that cell to be cancerous. So basically, cancer could simply be defined as the abnormality in the growth of cells. So they could be benign, where they don't spread to other sides of the body, or they could be malignant. And this is when the cancer cells spread from sites other than the primary site. The primary site is basically the site of where the cancer cell originated from. So metastasis is when cancer cells spread. That you could call that process metastasis. So it is the spread of cancerous cells to different parts of the body depending on the adhesion of cells to each other and the extracellular matrix. So if it's a cell-to-cell adhesion, or if it's an extracellular matrix adhesion. So cancer cells, they spread by invading or growing into normal tissues. So the only way that they can do that is if the cells are loosely attached to one another. So this is where our cell adhesion comes in place. So metastasis is dependent upon cell adhesion. So basically, the ability of a cell to actually spread to other parts of the body depends on how loosely the cells are in their interaction. So if cells that are adhere to one another are loosely attached to one another, it is easier for invasions or migrations to occur in that cell. So it's easier for cancer cells to spread into that to those cells. So you could just basically say that adhesiveness is said to be reduced between cancer cells. So now we're going to talk about epithelial to mesenchymal transition. So we know that epithelial tissues are polarized, meaning that they have different domains. They have the lateral domain, the apical domain, the basal domains, and they have different functions. So mesenchymal tissues are the opposites of epithelial tissues in the sense that they are not polarized. So they don't have those different domains. So if the cells that make up the mesenchymal tissues are loosely attached and lack the organization that the epithelial tissues have. So they're not as organized as regular epithelial tissues. So epithelial to mesenchymal transition involves the alteration between adhered cells so that the epithelial cells break down and become loosely attached to one another. When these cells are loosely attached to each other, it makes it actually easier for the cells to be invasive or migratory. The epithelial cells become invasive become invasive basically means that they are polarized, meaning that they have different domains. They have the lateral domain, the apical domain, the basal domains, and they have different functions. So mesenchymal tissues are the opposites of epithelial tissues in the sense that they are not polarized. So they don't have those different domains. So if the cells that make up the mesenchymal tissues are loosely attached and lack the organization that the epithelial tissues have. So they're not as organized as regular epithelial tissues. So epithelial to mesenchymal transition involves the alteration between adhered cells so that the epithelial cells break down and become loosely attached to one another. When these cells are loosely attached to each other, it makes it actually easier for the cells to be invasive or migratory. The epithelial cells becoming invasive basically means that these cells either lost their E cadherins partially or completely, because the most common cadherin found in epithelial cells are E cadherins. So if an epithelial cell is becoming invasive, it just basically means that it is the E cadherins are partially or completely lost. So because those are actually the cadherins that allow the cell adhesion. Those cadherins are the cell adhesion molecules that enable the cells to become more tightly attached to one another. So since it's already established that cancerous cells spread by migration and invasion to nearby normal tissues, epithelial to mesenchymal transition causes these said cells to become more prone to cancerous cells. So this epithelial to mesenchymal transition, it disrupts the cell junctions because they become loosely attached. So the cell junctions are not as functional as they were before, and then in this process you gain mesenchymal molecules such as N cadherins and fibronectin.
Next, we have the E-cadherin and the integrin crosstalk. So basically, the crosstalk between the E-cadherin and integrins plays a part in the cancerous tumor cells by responding to various signals in order to regulate migration and invasion. The crosstalk is mediated by the disruption of cell adhesion by the actin-myosin contractiolicity. So looking at the figure, signals such as the SRC and the focal adhesion kinase, integrin link kinase, and the small GTPases play a role in invasion, migration, and metastasis of tumor cells in the crosstalk. So the signal between SRC and Rho GTPases are pretty much connected. For example, SRC is able to inactivate Rho A integrin activation, whereas GTPases family can regulate the intercellular targeting of the SRC. As you can see here, that these are the downstream signal networks for the integrin. As you can see, it's transcription, actin remodeling, and so on. These signals can alter the balance between the adherent stability to non-migratory characteristics, or to adherent junction remodeling, which can lead to migration and invasion of epithelial cells.
So now we're going to briefly talk about dominant negative mutations. Previously, we had learned that there are different levels at which the expression of a gene can be regulated. So we have the genome level, we have transcriptional level, we have the post-transcriptional level, translational level, and the post. So RNA interference involves the regulation of gene expression at the post-transcriptional level, so after transcription occurs. So the short interfering RNA, also known as siRNA, help in the silencing of a particular gene at the post-transcriptional level. They do this by breaking down the transcribed mRNA, because we know that mRNA is produced in transcription. So, and they do this so as to silence the expression of that gene. So this happens because the RNA fragments bind to specific proteins that cause degradation of mRNA. So when you silence it, when you say you silence a gene, you're basically saying that you're not allowing a gene to be expressed. Another function of siRNA is to prevent mutation by binding to the promoter region of a targeted gene. So in an experiment to understand how cell adhesion affected siRNA efficiency, it was noticed that siRNA function improved with increased adherence. So you could see that siRNA, the silencing of a gene was more prominent when the cells were more attached to each other. The adherence of cells were more was increased. So siRNA prevents mutation that could possibly lead to cells becoming cancerous. And we will come back to this in a couple of seconds after we understand how this could affect dominant negative mutations. So a mutation is a change in the DNA sequence of a gene that leads to a different expression of that gene. So a dominant negative mutation is a mutation where a wild type allele or the original receptor molecule is altered to form a mutant that acts in opposition to the expression of the original receptor molecule. The expression of that mutant allele or receptor molecule dominates that of the wild type, and the wild type is the original allele or the original receptor molecule. So it acts in such a way that in the presence of the mutant, there is no function of the wild type allele. So P53 gene is a tumor suppressor. So it basically acts in such a way to suppress tumor by inhibiting or stopping cell growth when necessary to prevent cancerous cells from coming about. So the mutation of this gene can, however, cause a cell to become cancerous. So this is a dominant negative action. So if there's a mutant P53 gene in the presence of a wild type P53 gene, the wild type will not be able to perform its function because the mutant gene will not allow the wild type to actually bind to the gene promoter, so the gene is not expressed. So in the same way, we know that E-cadherin is also a tumor suppressor. And this E-cadherin is a protein, this protein is encoded by the CDH1 gene. So in a situation where there's a dominant negative mutation on this gene, the actual function of cadherin will cease to occur, and these cells will cease to adhere to each other as tightly as they are meant to be. So if you add a dominant negative mutation occurs in a E-cadherin protein, it could cause the E-cadherin to lose its function, and if it loses its function, the E-cadherin will not be expressed. And if it's not expressed, we know the E-cadherins help in the attachment of cells, so they could become closer together, so they could become less invasive and migratory, so we could so the cells could not be prone to becoming cancerous, to be allow cancerous cells to come with them. So if that happens, E-cadherins will not work, and the cells will actually become more prone to becoming more invasive, and cancer could be spread in that cell. So going back to understanding how DNA dominant negative mutation works, actually has a role on the siRNA on cancer. So we just saw that previously in the case of dominant negative mutation, we just saw the previous case of the dominant negative mutations. So if siRNA prevent mutations, as in the case of the P53 gene and the E-cadherin protein, which codes for the for a specific gene, which we said was CDH1, siRNA will prevent those mutations, and such that the E-cadherins are actually able to perform their functions as tumor suppressors. And when they do this, it would prevent the invasion of nearby tissues by cancerous cells. because the E cadherins would allow the adjoining of cells to attach to each other more tightly. So with this, we are able to conclude our video. We were able to see how cells adhere to each other, the different cell adhesion molecules, with giving more importance to the integrins and the cadherins. We understood how dominant negative mutations could affect cadherins. These cell adhesion molecules, with the example of most examples giving in the case of E cadherin, which is a tumor suppressor gene, and we also saw how these what role these cell adhesion molecules played in